Hello everyone, we are going to discuss what is ethnobotany, the role of ethnobotany in drug discovery and good manufacturing practices in herbal therapeutics. Why are these good manufacturing practices required in herbal therapeutics? How the application of good manufacturing practices change the quality of herbal therapeutics. Ethnobotany, which is a scientific study of how people use and relate to plants in their cultural tradition. So, we are aware that since time immemorial, humans have been using plants for food, shelter and different purposes, and this is how we have discovered the use of plants for different applications, may it be food, may it be medicine. And if we look at different cultures, especially in India, each cultural group is using specific group of plants either as nutraceuticals or to treat some of the diseases. So, studying this gives us a very important insight in developing a modern day dosage form. Ethnobotany plays a very vital role in herbal therapeutics by documenting indigenous knowledge, guiding drug discovery and supporting sustainable healthcare practices. The term ethnobotany was first introduced by American botanist John Harshbarger in 1896 as the study of plant use by humans. He closely observed human beings using different plants, and the first documentation came from him. So, ethnobotany is the study of plant human interrelationship embedded in a dynamic ecosystem of natural and social component. Ethnobotanists have a responsibility both to the scientific community and to the indigenous cultures. So, when they are trying to study respecting that culture and carefully documenting different uses of plants is a huge responsibility on these ethnobotanists. An ethnobotanical text evolves around the human community's use and management of these vegetations. So, the core meaning of ethnobotany is study of the relationship between human and plants, focusing on how different cultures identify, use and manage plant species. For example, in India itself, if you look at different traditions and cultures, there may be different plants used to treat acidity, different plants used for as antispasmodic, different plants used to treat some kind of pain. So, all this identification and documenting is under the purview of ethnobotany. The scope includes food, medicine, rituals, ecological practices, but its strongest impact has been in traditional medicine and herbal drug development. So, different rituals, especially in India, do use different kind of plants. This also gives us an important lead for drug discovery. Scientific relevance, ethnobotany bridges anthropology, botany and pharmacology, creating a knowledge base that connects traditional wisdom with modern medicine. What is the role of ethnobotany in herbal therapeutics? Most important is documentation of indigenous knowledge. It preserves the century old practices of plant used in healing. Else, today we would not have known these age old medicines which today are used as home remedies. It provides a systemic inventory of medicinal plants used by tribal and rural communities. It prevents the loss of traditional wisdom due to modernization. So, a geographical patent is very important when we know the traditional use of a medicine. That is how India could patent turmeric, since it is traditionally used in India. Drug discovery and development, where many modern day drugs are discovered, quinine, morphine, paclitaxel, which originated from ethnobotanical leads. The Ethnobotanical Survey also helps identify plants with bioactive compounds for further pharmacological testing. This supports translational research by linking folk remedies to validated therapeutic agents. The role in herbal therapeutics, phytochemical and pharmacological insights. Ethnobotany guides researchers towards plants and therapeutic potential, reducing the trial and error in drug screening. Else, imagine how much trial and error one needs to do in a drug discovery. Suppose you want to do a drug discovery, say in cancer or an antibiotic or hypertension, some lead from the traditional use will reduce so much of time and money which can be helpful in faster screening and drug discovery. Sustainable healthcare, where it promotes cost effective, culturally relevant therapies and encourages conservation of medicinal plants. So, we will not unnecessarily exploit different medicinal plants and biodiversity if there is a very important lead. It supports eco-friendly approaches to healthcare by reducing dependence on synthetic drug. There can be integration with modern medicine, and ethnobotany provides a strong foundation for integrative medicine, and hence we can convert the herbal medicine into modern day dosage forms with an evidence base. It strengthens the regulatory framework by offering ethnobotanical justification. Most of these drugs have stood the test of time and not shown toxicity. So, this becomes a very strong ethnobotanical justification. For example, turmeric, neem, tulsi, these have been used since time immemorial and have not shown any kind of toxicity. But there are some challenges which we should remember. The risk of biopiracy, indigenous knowledge can be exploited. There is a need for validation, enough scientific proof needs to be generated. There are conservation issues in over harvesting of medicinal plants and regulatory hurdles in translating exactly the ethnobotanical knowledge into modern therapeutics. So, ethnobotany is more than just cataloging plant uses. It is a strategic tool for herbal therapeutics, enabling discovery, validation and sustainable healthcare. So, by respecting the traditional wisdom while applying to modern science, ethnobotany ensures that herbal medicine remains both culturally rooted and scientifically credible. Let us look at some examples which are derived from an ethnobotanical lead. Quinine is a classic example which was discovered from cinchona tree, and this is today used as an anti-malarial. So, indigenous people of South America, this comes from Peru, used cinchona bark to treat fevers, and the water of cinchona bark was given as holy water by the priest to treat fevers of unknown origin. Later, the drug discovery led to identification of quinine as a very important constituent present in cinchona species. So, this became the first effective treatment for malaria, later inspiring synthetic derivatives for chloroquine, so that we do not harvest cinchona trees from wild sources. Paclitaxel, which was screened by NIH for anti-cancer activity, today has become a very important modern day lead, and this was obtained from a lead from the Native American tribes who used yew for medicinal purposes. Paclitaxel is a frontline chemotherapy drug today for ovarian, breast and lung cancers. Morphine, again, which is a classic example, obtained from a natural source. This is the opium capsule, and I mentioned earlier that incision is given only on the outer periphery, and this is the latex which is rich in alkaloids. The latex is collected, from which the alkaloids are later extracted. Morphine remains a gold standard for severe pain management, especially in terminally ill cancer patients. Artemisinin, which is an anti-malarial drug, obtained from Artemisia annua. This was documented in the Chinese medicine for treating fevers. So, these are some of the ethnobotanical leads which have led to modern drug discovery, and a combination of artemisinin based therapy is most effective today to treat the resistant malarial parasite. Rezapine, which is obtained from Rolfia serpentina. This is very much an Indian indigenous drug mentioned in Ayurveda texts, and this was used in Ayurveda for snake bites, for insomnia and mental illness. This root looks like a snake and hence the name serpentina, and the active constituents were first discovered by Dr. Rolf and hence the name Rolfia serpentina. The common name is Sarpagandha. Aspirin, a simple molecule which was first found in willow tree bark and used as commonly analgesic and anti-inflammatory, and this has led to development of acetyl salicylic acid, which is one of the most widely used drugs globally today. Why are these examples important? Because they reduce the trial error by pointing researchers towards plants with proven traditional uses. Drug discovery pipelines often start with ethnobotanical surveys, then move to phytochemical isolation and clinical validation, cultural respect and biodiversity conservation, and hence ethnobotany is one of the most important cornerstones of modern pharmacology. Good manufacturing practices, which goes hand in hand with processing, with ethnobotany and other aspects of herbal therapeutics. This has to be inculcated when we are formulating an herbal medicine, because these are internationally recognized standards that ensure herbal medicine are consistently produced, controlled and distributed with high quality, safety and efficacy. These cover everything from the raw material sourcing to packaging, storage and documentation till it finally reaches the end user or the consumer. What is this good manufacturing practice when we relate it to herbal therapeutics? It refers to a set of guidelines and regulatory standards that govern the manufacturing of herbal medicine, and there are several more steps involved here as compared to a synthetic medicine. The purpose is to guarantee that herbal products are free from contamination, they are properly standardized. Since they are obtained from nature, there can be a lot of variation when there are natural changes in climate or soil. So, it becomes very important to follow these good manufacturing practices when we are planning a herbal therapeutic. The World Health Organization has issued specific GMP guidelines, and these are available and must be followed for regulatory compliance, especially when we are planning an export of herbal therapeutics. The key components of GMP for herbal medicines is the raw material control, which we have little discussed in our earlier lesson, authentication, harvesting, testing, the premises and equipment where the pharmacovigilance is strict in the factory premises. Pharmacovigilance should also be done during cultivation, whether good agricultural practices are followed during cultivation and storage, personnel and training, especially whether they have knowledge of pharmacognosy and phytochemistry, the production practices where standardized extracts and formulation methods are used, batch records are maintained for traceability. Quality control must be maintained during each step of the formulation of herbal therapeutics. Documents must be properly maintained, and distribution and post market surveillance is also now applicable to herbal therapeutics. The importance of applying good manufacturing practices is to ensure safety, it guarantees efficacy, builds trust, supports regulation and promotes global trade. However, there are some challenges which we should keep in mind. The complexity of plant materials, since it is naturally obtained from nature and each plant is packed with several bioconstituents. Authentication issues, since there is a risk of misidentification, resource limitations and regulatory harmonization. These herbal medicines are regulated by Ayush, they are regulated by WHO, they are regulated by each traditional practitioner, hence we have to be very careful. GMP in herbal therapeutics is the backbone of quality assurance, ensuring that traditional remedies are transformed into reliable, safe and standardized medicine, and this provides a framework to bridge ethnobotanical knowledge with modern regulatory science. Let us see a few examples of leading herbal formulations which are there in the market today, consumed by a large population. A very well known hepatoprotective formulation based on ethnobotany or traditionally used herbs, which is used for liver disorder, is derived from plants like Capparis spinosa, Sycorium intubus, Solanum nigrum and Terminalia arjuna. So, the GMP features that the raw material authentication and phytochemical standardization must be done. At times it may be very difficult to do a phytochemical standardization on the final product, especially when it is a polyherbal formulation and has more than 25 or 30 herbs. So, in such cases it is better to incorporate a standardized extract. Controlled extraction processes to preserve the active constituent. So, here we ensure that the extract has got the minimum bioactive constituents in it, and quality checks for microbial contamination, heavy metals and pesticide residues. This can be done on the final formulation. So, the outcome is a consistent, safe, effective hepatoprotective herbal medicine accepted in global markets. Another classic Ayurvedic rasayana, an immunomodulator based on ethnobotany which uses amla. It combines multiple herbs with amla for regeneration and immunity. So, here it is very important to standardize this with respect to ascorbic acid and tannins, so that this immunomodulator can be accepted globally with a consistent GMP. A digestive blend very commonly found in the market, which has combination of 3 fruits, harda, beheda and amla, and this is used for digestion and detoxification. This involves botanical identification, since all these 3 fruits are rich in tannins. All of them contain stone cells if you see the powders under the microscope, but if you observe carefully, these stone cells are of different types. So, this can be used for botanical identification, hygienic processing and validated equipment. So, the ash values will give a very good control on the cleaning and processing of these fruits, and this reliable formulation will produce good expected therapeutic effects. For an anti-malarial formulation which consists a blend of ethnobotanical documented plants, here the GMP certified facilities with rigorous pharmacological validation and clinical trials. So, this is an example of a formulation where it is used as supportive or anti-malarial formulation. Why GMP matters? So, after looking at these examples, it is a proof of how the traditional ethnobotanical wisdom is transformed into modern standardized therapeutics. Hence, GMP is very important to ensure quality, safety and global acceptance of herbal medicines, and these formulations illustrate and bridge the heritage and knowledge with pharmaceutical credibility. So, this table summarizes how the raw material selection is done, processing is done, formulation design is done, and quality is maintained using ethnobotany and the modern features. Documentation is very important, along with packaging and storage, distribution and use, and the safety and efficacy maintained. Hence, concluding, because there is a lot of variability in herbal medicines, these plant based formulations differ in quality due to difference in geographical locations from where they are collected, seasonal variations, processing at different geographical locations, and this makes the standardization essential. Good manufacturing processes hence must be applied and ensure authenticity, purity and consistency by regulating sources, processing, quality control and documentation. The scientific and global necessity is a must for elevating these traditional remedies and converting them into credible, safe and effective therapeutics, making them globally accepted. Thank you.